Alzheimer's disease begins in absolute silence, no warning, no pain, just a single protein folding the wrong way in the dark. A microscopic betrayal that slowly, relentlessly unravels a human life from within. As humanity lives longer than ever before, a new enemy emerges, not airborne, not contagious, but born from the very machinery that creates memory and identity. Beta amyloid and tau turn rogue, building plaques and tangles that choke the brain's vast electrical empire. The first strike hits the memory gateway, the hippocampus falls next, thoughts fade, words slip away, familiar faces blur into shadows. Across the world, families stand helpless as someone they love slowly disappears while the body remains caregivers awake at dawn. Spouses holding hands with someone who no longer knows them. But science fights back, imaging, genetics, global research teams all racing to slow or stop the unmaking of the human mind. The complete science of Alzheimer's disease, where memory begins and where it falls. Before we begin, if you enjoy these types of interesting science topics, hit the like button, comment, and tell us where you are listening from. If you'd like to hear more stories like this, you know what to do. Simply subscribe. In the vast timeline of human existence, Alzheimer's disease is a relatively recent revelation, not because it appeared suddenly, but because humanity had to live long enough to discover it. For most of our species' history, life was brief. Our ancestors who survived childhood rarely reach the ages when this disease emerges from the shadows of the aging brain. The origins of Alzheimer's lie not in an external pathogen, not in a virus carried by wind or water, but in the intricate machinery of our own cells, machinery that, given enough time, begins to falter. The story begins with proteins, those molecular workers that build and maintain every structure in our bodies. In the landscape of the human brain, proteins fold into precise shapes, each configuration enabling specific functions. They transport nutrients, transmit signals, repair damage, and maintain the delicate architecture of our thoughts and memories. But proteins are not immortal. They wear out, become damaged, accumulate errors. In a healthy brain, sophisticated cleanup systems patrol constantly, identifying misshapen proteins and breaking them down before they cause harm. Alzheimer's emerges when this cleanup system fails. Two proteins in particular, beta amyloid and tau, become the architects of destruction. Beta amyloid, normally a short fragment that gets cleared away, begins to accumulate. These fragments stick together, forming clumps outside brain cells, creating structures called plaques. Meanwhile, tau proteins, which normally stabilize the internal scaffolding of neurons, become chemically altered. They detach from their supportive roles and twist into tangled filaments inside cells, choking the neuron from within. This process is not a sudden invasion. It is a slow betrayal by the body's own molecules. The plaques and tangles do not appear overnight. They accumulate gradually, silently, over decades. By the time a person notices the first slip in memory, the first struggle to find a familiar word, the disease has already been working in darkness for perhaps 20 years or more. The evolutionary perspective reveals a paradox. In the deep past, when few humans lived beyond their fourth or fifth decade, Alzheimer's remained hidden. Natural selection, which shapes our biology through survival and reproduction, exerts little pressure on what happens after reproductive years end. The genetic variations that might predispose someone to Alzheimer's pass through generations unchallenged because their bearers typically died of other causes before. The disease could manifest as human civilization advanced, as we developed agriculture, sanitation, medicine, and social structures that protected the vulnerable. Our lifespans extended. We began routinely reaching our 60s, 70s, 80s, and beyond. This extended longevity, one of humanity's greatest achievements, also unveiled diseases that had always been part of our biology but rarely had time to develop. Alzheimer's is, in this sense, a disease of success, revealed only because we learned to live long enough to experience it. The earliest cases likely occurred throughout human history, but they blended into the general decline attributed to old age. Ancient texts occasionally mention elders who lost their memories, who no longer recognized family, who wandered in confusion. But these observations remained scattered, anecdotal, unconnected to a specific disease process. The modern recognition of Alzheimer's as a distinct condition would not emerge until medicine developed tools to look inside the living brain, to examine tissue under microscopes, to distinguish different types of cognitive decline. The geographic origins of Alzheimer's are universal. This is not a disease that arose in one location and spread to others. It exists in all human populations, across all continents, in every culture. What varies is not the disease itself, but awareness of it, access to diagnosis, and the social structures that determine how societies understand and respond to cognitive decline in their elders. In this way, Alzheimer's represents a fundamental challenge of being human, a consequence of the complexity that makes us who we are. Our brains, vast networks of nearly 100 billion neurons, making trillions of connections, are perhaps the most intricate structures in the known universe. This complexity enables consciousness, creativity, love, and memory. But complexity also creates vulnerability. The more intricate the system, the more ways it can fail. Alzheimer's emerges from the intersection of our biological sophistication and the simple reality that all systems, given enough time, accumulate damage faster than they can repair it. The formation of Alzheimer's disease begins in the realm of the molecular, in processes so small they remained invisible until modern science developed the tools to observe them. Understanding how this disease begins requires understanding what happens when proteins, the fundamental building blocks of cellular function, lose their proper shape and purpose. Every cell in the human body constantly produces proteins. These molecules are created based on genetic instructions, emerging as long chains of amino acids that must fold into specific three-dimensional shapes to function correctly. The shape of a protein determines its role, like a key fitting a lock, like a tool designed for a particular task. In brain cells, some proteins form channels that allow chemical signals to pass. Others provide structural support. Still others act as cleanup crews, breaking down waste products. Beta-amyloid protein begins as a larger molecule called amyloid precursor protein, embedded in the membrane of neurons. Normally, enzymes snip this precursor protein at specific locations, creating fragments that dissolve harmlessly into the fluid surrounding brain cells. But sometimes, the cutting happens at slightly different points, producing a longer, stickier fragment. This longer version of beta-amyloid has a dangerous tendency. It clumps together. At first, these clumps are small, just a few molecules adhering to each other in the spaces between neurons. These tiny clusters, called oligomers, may be the most toxic form. They interfere with communication between brain cells, disrupting the precise chemical conversations that encode thoughts and memories. As more beta-amyloid accumulates, these small clumps grow into larger deposits called plaques, dense collections of protein that sit outside neurons like debris accumulating in a once clean stream. The question of why this accumulation occurs has no single answer. In some cases, genetics plays a clear role. Certain inherited genetic variations directly affect beta-amyloid production or clearance. People who inherit these variations may develop early onset forms of the disease, sometimes in their 40s or 50s. But these genetic cases represent a small fraction of all Alzheimer's disease. Most cases occur in people with no strong family history, suggesting that multiple factors contribute. Age itself is the greatest risk factor. As the brain ages, its cleanup systems become less efficient. The enzymes that normally clear away beta amyloid work more slowly. The cellular machinery that recognizes and removes damaged proteins becomes overwhelmed. Inflammation increases throughout the brain, possibly in response to a lifetime of minor injuries, infections, and metabolic stress. This chronic, low-level inflammation may further impair protein clearance and even promote beta amyloid production. Meanwhile, tau protein begins its own destructive transformation. Normally, tau proteins stabilize microtubules, the internal railway system that transports nutrients and chemical signals along the length of neurons, which can extend remarkable distances in the human brain. Some neurons stretch from the spinal cord to the toes, and even within the brain itself, neurons connect across significant distances. The microtubules need constant stabilization, and tau provides it. But in Alzheimer's disease, tau proteins become chemically modified through a process called hyperphosphorylation. Extra phosphate molecules attach to the tau, changing its properties. Modified tau detaches from microtubules and begins sticking to other tau molecules, forming twisted filaments. These tangles grow inside neurons, choking the cell from within. The internal transportation system collapses. Nutrients cannot reach the distant parts of the neuron. Waste products accumulate. Eventually, the neuron dies. The relationship between beta amyloid plaques and tau tangles remains a subject of intense research. Current evidence suggests that beta amyloid accumulation may occur first, creating conditions that trigger tau pathology. The plaques outside neurons may stress the cells, triggering inflammatory responses and metabolic changes that promote tau modification. Once tau tangles begin forming, they spread through connected networks of neurons in a pattern that correlates closely with symptom progression. Other factors influence disease formation. Cardiovascular health affects Alzheimer's risk. What is good for the heart is often good for the brain. High blood pressure, diabetes, and high cholesterol in midlife all increase later risk of dementia. The brain requires enormous amounts of energy and oxygen delivered through an intricate network of blood vessels. Anything that damages these vessels or reduces blood flow can make neurons more vulnerable to the stresses of aging and protein accumulation. Head injuries, particularly repeated impacts or severe trauma, also increase risk. Such injuries may trigger inflammatory processes that persist for years, or they may damage the brain's drainage systems that normally clear away waste proteins. Sleep quality matters too. During deep sleep, the brain clears metabolic waste through a recently discovered system of channels that flush away accumulated debris. Chronic poor sleep may allow waste products, including beta amyloid, to accumulate. Education, cognitive engagement, and social connection appear to offer some protection, though they do not prevent the disease entirely. The concept of cognitive reserve suggests that brains with more robust neural networks can better tolerate damage before symptoms emerge. A lifetime of learning builds redundancy into neural systems, more pathways to accomplish the same cognitive tasks. When Alzheimer's begins destroying some pathways, alternative routes remain functional longer. Yet even with all these contributing factors understood, the fundamental question remains mysterious. Why do some people develop dense accumulations of plaques and tangles while others with similar risk factors do not? Why does the disease begin at different ages in different people? The answer likely involves complex interactions between genetics, environment, immune function, and perhaps elements of chance and cellular processes that unfold over decades. Alzheimer's disease does not strike the brain uniformly. It follows patterns, spreading through neural networks in a sequence that reveals itself through the gradual loss of different cognitive abilities. Understanding this progression means understanding the geography of the brain, which regions control which functions, and why the disease affects some areas before others. The spreading begins in a region called the entorhinal cortex, a small area deep within the temporal lobe. This region serves as a gateway to the hippocampus, the brain's memory-forming center. Information from across the brain funnels through the entorhinal cortex to reach the hippocampus, where experiences transform into memories. When tau tangles first appear in significant numbers, they appear here, in this crucial bottleneck of memory formation. From the entorhinal cortex, the pathology spreads to the hippocampus itself. The hippocampus, shaped like a seahorse, lies deep within each temporal lobe. It binds together different aspects of experience, the sights, sounds, emotions, and context of a moment into cohesive memories. As tangles accumulate in the hippocampus, the ability to form new memories begins to fail. This explains why early Alzheimer's primarily affects recent memory while leaving older memories relatively intact. The hippocampus creates new memories, but older memories have already been transferred to other brain regions for long-term storage. The disease then extends into adjacent temporal lobe regions involved in language and object recognition. The ability to name objects declines. Words become elusive, dancing just out of reach. A person might describe an object's function without being able to produce its name, saying, "the thing you write with," instead of pen. These difficulties reflect damage to regions that connect words with meanings, that retrieve linguistic information on demand. As the disease progresses, tau tangles and beta amyloid plaques spread more widely across the cortex, the folded outer layer of the brain where higher-order thinking occurs. The parietal lobe, which integrates sensory information and helps us navigate space, becomes affected. People lose the ability to judge distances, struggle to navigate familiar routes, and may become disoriented even in their own homes. The brain's internal map of the world begins to dissolve. The frontal lobe, sitting behind the forehead and governing executive function, planning, and personality, also sustains damage. As this region deteriorates, people lose the ability to plan sequences of actions, to solve problems, to make sound judgments. Personality changes may emerge. A formerly cautious person might become impulsive. A gentle person might display uncharacteristic anger or frustration. These changes are not choices, but reflect the physical destruction of the neural circuits that regulate behavior and emotion. The visual processing areas in the occipital lobe can also be affected, though often later in the disease course. This can cause difficulties recognizing faces, even of close family members, or interpreting complex visual scenes. Hallucinations may occur, particularly in dim lighting, as the damaged brain struggles to construct coherent visual interpretations from ambiguous sensory input. Throughout this spreading process, neurons die. The brain physically shrinks as cells disappear and the spaces filled with cerebrospinal fluid expand. Brain imaging reveals this atrophy, showing enlarged ventricles, the fluid-filled cavities inside the brain, and widened gaps between the folded ridges of the cortex. The physical loss of tissue corresponds directly to the loss of function. Each region that atrophies takes with it the abilities it once enabled. The mechanism of spread appears to involve a disturbing process. Misfolded tau proteins may transfer from cell to cell along connected pathways. Neurons that connect to each other, that communicate regularly through synapses, may pass these toxic proteins between them. The tau spreads not randomly but along anatomical pathways, following the wiring diagram of the brain. This pattern explains why damage progresses in relatively predictable stages, moving through connected networks rather than appearing simultaneously everywhere. Beta amyloid plaques accumulate more diffusely, but still show regional patterns. They appear widely across the cortex and seem to create an environment that promotes tau pathology. Current research suggests a two-hit model. Beta amyloid accumulation makes neurons vulnerable, and tau tangles deliver the fatal blow. Regions with both pathologies deteriorate fastest. Inflammation accompanies this spread. The brain's immune cells, called microglia, respond to the accumulating plaques and dying neurons. These cells attempt to clear debris and protect surviving neurons, but chronic activation of microglia may cause collateral damage. They release inflammatory molecules that, over time, harm nearby healthy neurons. What begins as a protective response becomes part of the disease process itself. Blood vessels in the brain also deteriorate. The blood-brain barrier, which normally prevents harmful substances from entering brain tissue, becomes leaky. This may allow inflammatory factors from the bloodstream to enter the brain. Blood flow decreases in affected regions, depriving neurons of oxygen and glucose. Some plaques even form in blood vessel walls, further compromising circulation. The spreading process takes years, even decades. Early stages may be nearly silent, with pathology present but compensation mechanisms still effective. Middle stages bring noticeable symptoms that progressively worsen. Late stages involve widespread brain damage, profound cognitive impairment, and eventually the loss of basic functions, like swallowing and maintaining blood pressure. The journey from first molecular changes to end-stage disease typically spans 20 to 30 years, though the pace varies considerably among individuals. The history of Alzheimer's disease is not a story of geographic spread, like a plague moving from city to city, but rather a story of recognition of humanity, gradually distinguishing this specific form of dementia from the general haziness attributed to old age. The disease has always been part of the human condition, hidden within populations, affecting elders whose confusion and memory loss were considered inevitable consequences of aging. Ancient texts occasionally mention cognitive decline in the elderly. Egyptian papyri from thousands of years ago reference old age and its associated difficulties. Greek and Roman physicians described senility, though without distinguishing between different causes. Chinese medical texts acknowledge that some elders lost their minds, while others remained sharp, but these observations remain descriptive, not diagnostic. No one imagined that specific disease processes might underlie these changes. The modern medical recognition began in the early 1900s in Frankfurt, Germany. A physician named Alois Alzheimer examined a patient who developed severe memory problems, paranoia, and confusion in her early 50s. After her death, he examined her brain tissue under a microscope and observed unusual features: plaques scattered throughout the tissue, and strange tangles inside neurons. He presented his findings to the medical community, describing what he believed was a distinct disease process. For decades after this initial description, the condition remained rare in medical literature, not because it was truly rare, but because most cases occurred in elderly people whose symptoms were attributed simply to age. The term gradually entered medical vocabulary, but primarily for cases occurring before old age. Only later did researchers recognize that the same pathological features appeared in much more common late-life dementia. The mid-20th century brought gradual shifts in understanding. As populations in industrialized nations aged, the number of people living into their 70s and 80s increased dramatically. The prevalence of dementia became more apparent, and it became less acceptable to dismiss profound cognitive decline as normal aging. Researchers began systematically studying older adults, conducting autopsies, examining brain tissue, and correlating pathological findings with clinical symptoms. By the late 20th century, Alzheimer's disease emerged as a major public health concern. Improved diagnostic criteria allowed doctors to identify probable cases during life rather than only through autopsy. Brain imaging technologies enabled visualization of atrophy and metabolic changes. Awareness increased among both medical professionals and the general public. Advocacy organizations formed, Research funding expanded. The disease that had been nearly invisible became recognized as one of the most significant health challenges of aging societies. Different regions of the world have experienced varying trajectories in confronting Alzheimer's. Wealthy nations with advanced healthcare systems developed diagnostic infrastructure, memory clinics, specialized care facilities, and research programs. Nations with younger population demographics faced less immediate pressure, but knew that aging populations would eventually bring increased dementia prevalence. Lower-income regions struggled with limited resources, where diagnostic capacity might be minimal, and where families bear the burden of care with little external support. Cultural factors shape how Alzheimer's is perceived and managed. In some societies, caring for elderly parents at home is a deep cultural obligation, and placing a relative in institutional care carries stigma. In others, nursing homes and memory care facilities are accepted solutions. Some cultures view cognitive decline as shameful, something to hide from neighbors and community, while others speak openly about the challenges. These cultural differences affect diagnosis rates, care patterns, and the lived experience of patients and families. The global distribution of Alzheimer's follows population aging. As birth rates decline and life expectancy increases worldwide, the proportion of elderly people grows. This demographic transition, most advanced in Europe, Japan, and North America, is proceeding rapidly in China, Latin America, and parts of Asia. Africa and the Middle East, with younger populations currently, will face similar transitions in coming decades. The global burden of dementia increases steadily, with projections suggesting the number of affected individuals will triple over the next several decades if no preventive treatments emerge. Public health responses vary enormously. Some nations have developed comprehensive dementia strategies involving early detection, support services, caregiver assistance, and research investment. Others lack national plans, leaving families to navigate the disease with minimal guidance. Access to medication, brain imaging, and specialized care differs by orders of magnitude between wealthy and poor regions, between urban and rural areas, between insured and uninsured populations. Historical approaches to caring for people with severe dementia were often inadequate or cruel. Before modern understanding, confused elders might be confined, institutionalized in poor conditions, or simply left to wander. The development of person-centered care approaches, which emphasize dignity and quality of life even in severe disease stages, represents significant ethical progress. Yet implementation of such approaches remains uneven globally. Research collaborations increasingly cross national boundaries. Large studies now track thousands of individuals across multiple countries, pooling data to identify risk factors and test interventions. Genetic studies involve tens of thousands of participants from diverse ancestries. Brain imaging repositories contain scans from around the world. This global scientific enterprise recognizes that Alzheimer's is a universal human challenge, requiring collective effort. The appearance of Alzheimer's disease in modern life begins subtly, often so gradually that its presence remains ambiguous for months or years. The first signs typically involve memory, not the occasional forgotten name that everyone experiences, but a persistent pattern of forgetting recent events and conversations. A person might ask the same question repeatedly, unaware they already received an answer minutes earlier. They might struggle to recall what they ate for breakfast or whether they took their medication. These early memory slips often provoke anxiety in the affected person, who notices something is wrong but may not understand what. They might compensate by writing extensive notes, by relying more heavily on routines, by asking family members to remind them of appointments. Some withdraw from social situations where their difficulties might become apparent, declining invitations, avoiding complex conversations, staying home more often. As the disease progresses, The memory problems become more pronounced and expand beyond mere forgetfulness. Entire conversations disappear from memory. Planned activities are forgotten. People may become lost driving routes they have traveled for decades, unable to recognize familiar landmarks or remember directions. Time becomes confused. They might dress for an appointment that is not until next week, or believe they recently saw someone who died years ago. Language difficulties emerge alongside memory problems. Word finding becomes harder. Sentences trail off mid-thought because the speaker cannot locate the words to complete the idea. Following complex conversations becomes difficult, as the brain struggles to hold multiple threads of meaning simultaneously. Reading comprehension declines, with the end of a sentence forgotten by the time the beginning was. Writing becomes simpler, sentences shorter, vocabulary more limited. In different regions of the world, these symptoms manifest within vastly different contexts. In wealthy urban areas with robust healthcare infrastructure, these changes might prompt medical consultation, neuropsychological testing, brain imaging, and eventual diagnosis. In rural areas or regions with limited medical access, the same symptoms might be interpreted as normal aging, with no formal diagnosis ever made. The experience of having Alzheimer's thus varies not just with disease severity but with social and economic circumstances. In cultures with multi-generational households, family members may notice changes first. Adult children living with aging parents observe the forgotten conversations, the repeated questions, the growing confusion. They might initially attribute these changes to stress or distraction, only gradually recognizing a pattern. In societies where elderly people live independently, isolation can mask early symptoms. Living alone, with no one to notice the daily slips, a person might develop significant cognitive impairment before others become aware. The middle stages of disease bring more visible difficulties. People require help with tasks that were once automatic, managing finances, cooking meals, selecting appropriate clothing for weather conditions. They might wear the same outfit repeatedly, forget to bathe, or neglect household maintenance. Judgment becomes impaired, leading to poor decisions, falling for scams, giving money to strangers, driving when no longer safe. Behavioral changes often emerge in middle stages. Someone who was always calm might become easily agitated or angry. Paranoia may develop, with accusations that family members are stealing possessions or plotting harm. These accusations, hurtful to family members, reflect the brain's attempt to explain missing items or confusing situations. Rather than remembering where an object was placed, the damaged brain constructs an explanation: someone must have taken it. Mood disturbances frequently accompany cognitive decline. Depression affects many people with Alzheimer's, possibly reflecting both brain chemistry changes and psychological response to declining abilities. Anxiety increases, perhaps because the world becomes confusing and unpredictable when memory and reasoning fail. Apathy, a profound loss of motivation and interest, can make people seem uncaring about things that once mattered deeply. Sleep patterns often deteriorate. The brain's regulation of sleep-wake cycles becomes disrupted, leading to nocturnal wandering, daytime drowsiness, or a phenomenon called sundowning, where confusion and agitation worsen in late afternoon and evening. For caregivers, these sleep disturbances can be exhausting, as they must remain vigilant through the night. In advanced stages, the disease affects nearly all cognitive domains. Speech becomes limited or absent. Mobility declines as the brain loses ability to coordinate movement. People require total assistance with dressing, bathing, toileting, and eating. They may no longer recognize close family members, though fleeting moments of recognition can occur. The personality that defined them seems to dissolve, leaving family members grieving someone who is physically present but psychologically absent. Physical complications emerge in late-stage disease. Difficulty swallowing leads to risk of choking or aspiration, where food or liquid enters the lungs. Immobility causes pressure ulcers, muscle wasting, and stiffening joints. Incontinence occurs as brain control of bladder and bowel function fails. Vulnerability to infections increases, with pneumonia often the final cause of death. Healthcare systems struggle to provide adequate care for people with advanced Alzheimer's. Memory care facilities specialize in dementia care but vary enormously in quality and cost. In many countries, such facilities are unaffordable for most families, who then provide care at home with limited support. Hospice and palliative care approaches, which prioritize comfort and dignity in terminal illness, are increasingly applied to end-stage dementia. Though cultural attitudes about such care vary, the public health infrastructure for Alzheimer's includes diagnostic services, support groups for patients and caregivers, respite care programs, and educational resources. Yet these resources are unevenly distributed. Even in wealthy nations, rural areas often lack specialized services. In developing regions, such infrastructure may be entirely absent, with families bearing full responsibility for care. Stigma remains a significant challenge. Despite increased awareness, many people fear an Alzheimer's diagnosis, delay seeking help, or hide symptoms from others. This stigma can isolate affected individuals and families, preventing them from accessing available resources and support. Public education campaigns aim to reduce stigma, emphasizing that dementia is a medical condition, not a personal failing or normal aging. Inside the brain of a person with Alzheimer's disease, a complex cascade of molecular and cellular changes unfolds over years and decades. Understanding this internal landscape requires traveling through scales from whole brain changes visible on imaging to molecular processes occurring inside individual neurons. At the largest scale, the brain physically shrinks. In advanced disease, the loss of tissue is dramatic enough to see on brain scans, or even in the weight and appearance of the organ at autopsy. The cortex, that wrinkled outer layer packed with neurons, thins. The hippocampus, seahorse-shaped and crucial for memory, atrophies severely. Fluid-filled spaces called ventricles expand as surrounding tissue disappears. This gross atrophy reflects the death of billions of neurons and the loss of the trillions of connections between them. Zooming into the tissue level reveals the pathological hallmarks, plaques and tangles. The plaques sit in the spaces between neurons, deposits of beta-amyloid protein that have aggregated into insoluble clumps. Under a microscope, they appear as dense spots scattered throughout the brain tissue, often surrounded by damaged neuron endings and activated immune cells. These plaques physically disrupt the local environment, interfering with normal cell-to-cell communication and triggering inflammatory responses. The tangles reside inside neurons, twisted filaments of tau protein that fill the cell body and extend into the cellular projections. These tangles choke the neuron from within, blocking the internal transportation system. Imagine a highway system where debris accumulates on the roads, first slowing traffic, then blocking it entirely. Nutrients cannot reach distant parts of the neuron. Waste products accumulate. Communication between the neuron and its neighbors breaks down. Eventually, the cell dies. At the synapse, the tiny gap where one neuron communicates with another, Alzheimer's wreaks havoc. Neurons communicate by releasing chemical messengers, called neurotransmitters, that cross the synapse and trigger responses in the receiving neuron. In Alzheimer's, synapses disappear. The complex molecular machinery that enables this communication disintegrates. Loss of synapses correlates more closely with cognitive decline than the number of plaques or tangles, suggesting that loss of connectivity matters more than the pathological deposits themselves. One neurotransmitter particularly affected is acetylcholine, which plays crucial roles in attention, learning, and memory. Alzheimer's destroys neurons in brain regions that produce acetylcholine. leading to deficits throughout the brain regions that depend on this chemical signal. Many Alzheimer's medications attempt to boost acetylcholine levels, temporarily compensating for this loss, though they cannot stop the underlying disease process. The brain's immune system participates actively in the disease. Microglia, the resident immune cells of the brain, normally patrol for damage, clear debris, and help maintain healthy neural tissue. In Alzheimer's, microglia become chronically activated in response to plaques, tangles, and dying neurons. Initially, this activation may be protective, with microglia attempting to clear away beta amyloid in damaged cells. But chronic activation transforms microglia into sources of damage themselves. Activated microglia release inflammatory molecules, chemical signals that promote inflammation. In acute situations, like fighting infection, inflammation is protective, but chronic, low-level inflammation damages healthy tissue. These inflammatory signals can make neurons more vulnerable to stress, can promote further protein misfolding, and can impair the brain's cleanup systems. The inflammation becomes self-perpetuating, creating a toxic environment even in regions without plaques or tangles. Another type of brain cell, called astrocytes, also responds to the disease. Astrocytes normally support neurons by regulating chemical environments, maintaining blood flow, and helping with debris clearance. In Alzheimer's, astrocytes become reactive, changing their shape and function. Like microglia, reactive astrocytes can release inflammatory signals. They may also lose their supportive functions, failing to provide neurons with necessary nutrients and failing to clean up neurotransmitters from synapses. Blood vessels throughout the brain deteriorate in Alzheimer's. The blood-brain barrier, a selective boundary that controls what enters brain tissue from the bloodstream, becomes leaky. This allows immune cells and inflammatory molecules from the blood to enter the brain, potentially amplifying inflammation. Some beta amyloid deposits form in blood vessel walls, a condition called cerebral amyloid angiopathy, which weakens vessels and can cause small hemorrhages or strokes. Blood flow itself decreases in affected brain regions. Positron emission tomography scans, which measure metabolic activity, show reduced glucose metabolism in Alzheimer's. The brain cells are not consuming energy normally. This could reflect decreased activity of damaged neurons, or it could indicate that neurons cannot access energy effectively, despite adequate blood flow. Either way, the energy crisis contributes to neuronal dysfunction and death. Mitochondria, the cellular powerhouses that generate energy, become dysfunctional in Alzheimer's. These small structures inside cells convert nutrients into the energy molecules that power all cellular processes. In Alzheimer's, mitochondria become damaged, possibly by oxidative stress, damaging molecules that accumulate when cellular chemistry goes awry. Impaired mitochondria mean neurons cannot generate adequate energy, making them vulnerable to the additional stresses of plaques, tangles, and inflammation. Oxidative stress itself increases in Alzheimer's brains. This involves reactive molecules that damage proteins, lipids, and DNA. Normally, cells have antioxidant defenses that neutralize these reactive molecules. In aging and disease, the balance tips toward oxidative damage. This damage can affect virtually any cellular component, from the membrane that surrounds the cell to the DNA in the nucleus, contributing to widespread cellular dysfunction. The systems that normally clear away protein debris become overwhelmed. Cells have sophisticated machinery for identifying damaged or misfolded proteins and breaking them down into components for reuse. This includes the proteasome, a molecular machine that chops proteins into fragments, and autophagy, a process where cells digest their own damaged components. In Alzheimer's, these clearance systems function poorly, whether due to aging, genetic factors, or the sheer quantity of misfolded protein they must process. The brain also has a waste clearance system that operates during sleep, recently discovered and called the glymphatic system. Cerebrospinal fluid flows through channels in the brain, flushing away metabolic waste, including beta amyloid. Disruption of this system, through poor sleep quality or other factors, might contribute to protein accumulation. The complex interplay between sleep, waste clearance, and Alzheimer's pathology represents an active research frontier. Genetic factors influence all these processes. Carrying certain genetic variations affects beta amyloid production, tau behavior, inflammatory responses, cholesterol metabolism, and immune function in the brain. The most significant genetic risk factor in common late-onset Alzheimer's involves a gene that affects how the brain handles cholesterol and responds to injury. Having certain versions of this gene substantially increases risk, though does not guarantee disease development. Throughout all these changes, the brain attempts compensation. Remaining healthy neurons may sprout new connections, trying to maintain function despite damage. Some brain regions may increase activity, attempting to take over for failing areas. These compensatory mechanisms explain why disease pathology can be present for years before symptoms appear. The brain is adapting, working around the damage. Eventually, though, the destruction outpaces compensation and function collapses. The long-term impact of Alzheimer's disease extends far beyond the individual patient, radiating outward to families, communities, healthcare systems, and entire economies. This disease does not simply end a life. It gradually erases identity while the body persists, creating unique challenges and profound suffering for all involved. For the person with Alzheimer's, the long-term trajectory involves progressive loss of self. In early stages, awareness of decline can bring depression, anxiety, and grief over the future being lost. As disease advances, awareness itself may fade, creating a paradox. The person becomes less distressed, precisely because they can no longer understand their situation. Philosophers and ethicists debate what it means to lose cognitive function, while the body continues, whether the person remains fully themselves, whether identity persists without memory and recognition. Quality of life becomes a central but challenging question. What constitutes good quality of life for someone who cannot express preferences, who may not remember experiences moments after they occur? Caregivers and medical professionals must infer needs and desires, making decisions on behalf of someone who cannot participate in those decisions. This requires balancing safety with autonomy, protection with dignity, medical interventions with comfort. For family caregivers, the burden is often described as one of the most difficult experiences a person can face. Caring for someone with advanced dementia involves constant vigilance, physical labor, emotional strain, and financial stress. The person being cared for may not recognize the caregiver, may resist help, may become combative or accusatory. The caregiver must repeatedly experience the grief of watching a loved one disappear while simultaneously managing the practical demands of round-the-clock care. Physical demands alone can be overwhelming. Helping someone dress, bathe, use the toilet, and move safely requires strength and patience. Sleep deprivation affects caregivers whose loved ones wander at night or have disrupted sleep-wake cycles. Caregivers frequently develop health problems themselves: hypertension, depression, weakened immune systems from chronic stress and exhaustion. Emotional and psychological impacts may be even more profound. The relationship between caregiver and patient fundamentally changes. An adult child caring for a parent with Alzheimer's experiences role reversal, becoming the parent to their own parent. Spouses watch their partner of decades become someone unrecognizable. The person they knew disappears while the body remains, creating a form of grief called ambiguous loss, mourning someone who is simultaneously present and absent. Social isolation frequently affects caregivers. The demands of care leave little time for friendships, hobbies, or social activities. Friends may withdraw, uncomfortable with the patient's condition or unsure how to help. Support groups can provide connection with others facing similar challenges, but many caregivers lack even the time to attend support groups. The isolation compounds stress and depression. Financial impact can be catastrophic. In countries without comprehensive social support for dementia care, families bear enormous costs. Memory care facilities can cost thousands per month, quickly depleting savings. Many families cannot afford such care and instead provide it themselves, often requiring someone to leave employment. Lost wages combined with out-of-pocket medical expenses create financial crisis. Even in countries with better social support, gaps in coverage leave families struggling. Healthcare systems face mounting challenges from the growing dementia population. Hospitals and emergency rooms see increasing numbers of people with advanced dementia experiencing acute medical problems. These patients often have difficulty communicating symptoms, may become agitated in unfamiliar hospital environments, and may not understand or cooperate with treatments. Traditional medical approaches designed for cognitively intact patients work poorly, yet alternatives remain underdeveloped. Long-term care facilities struggle to meet demand. In many regions, waiting lists for memory care beds stretch for months or years. The quality of institutional care varies enormously. The best facilities provide person-centered care, specialized training for staff, appropriate environmental design, and attention to dignity and quality of life. Many facilities, however, provide custodial care focused primarily on safety and basic physical needs, with little attention to cognitive engagement or emotional well-being. Staff caring for people with dementia, whether in facilities or as home care workers, often receive inadequate training and poor compensation despite the demands of the work. High turnover rates result, meaning people with dementia experience constant changes in caregivers, which can increase confusion and distress. The undervaluation of care work reflects broader societal attitudes but has direct consequences for people with dementia. The global burden of dementia falls disproportionately on low and middle-income countries, which bear the majority of cases but lack resources for adequate care. Extended families traditionally provide care, but urbanization, changing family structures, and economic pressures make this increasingly difficult. Without formal support systems, families in these regions face impossible choices between earning a living and caring for disabled relatives. Gender disparities characterize the dementia burden. Women develop Alzheimer's more frequently than men, partly because they live longer on average. Women also provide the majority of unpaid caregiving, often sacrificing careers and their own health to care for husbands, parents, or in-laws. This creates economic disadvantage that extends into their own old age, as interrupted careers mean lower lifetime earnings and reduced retirement savings. Comorbid medical conditions complicate care in later stages. People with advanced dementia may develop cancer, heart disease, infections, or other illnesses requiring medical decisions. Determining appropriate treatment becomes ethically complex. Aggressive medical interventions may prolong life without improving quality. Families must decide about hospitalizations, surgeries, feeding tubes, and resuscitation decisions made more difficult because the person with dementia cannot express preferences. End-of-life care presents particular challenges. Dementia typically progresses to death over many years, making prognosis uncertain and timing of transitions to hospice care difficult. Some cultures and healthcare systems embrace palliative and hospice approaches that prioritize comfort, while others default to aggressive medical intervention until death. Advanced care planning, where people express wishes about future medical care while still capable, remains underutilized despite its importance. After death, family members often experience complicated grief. They may have been grieving incrementally for years as the person gradually disappeared. When death finally comes, it may bring relief mixed with guilt about feeling relieved. The years of caregiving may have strained family relationships, created financial hardship, and taken physical and emotional tolls that persist long after the loved one dies. Research into interventions focuses not only on disease modification but also on improving quality of life and reducing caregiver burden. Non-pharmacological approaches, structured activities, music therapy, reminiscence therapy, environmental modifications. Caregiver education and support can improve outcomes for both patients and caregivers. Yet these interventions require resources and expertise that remain inaccessible in many settings. The societal conversation about Alzheimer's increasingly emphasizes the concept of dementia-friendly communities environments designed to support people with cognitive impairment remaining engaged in community life. This involves education to reduce stigma, environmental modifications to improve safety and navigation, and training for service workers in stores, banks, and public spaces to interact effectively with people with dementia. Such approaches recognize that quality of life depends not only on medical care, but on social inclusion. Alzheimer's disease occupies a unique position in the human story, a condition that emerges from our own biology, revealed by our success in extending lifespans, challenging our societies to care for those who have lost the very capacities that make us human. The story of Alzheimer's is simultaneously a story of human vulnerability and human resilience, of scientific progress and humbling limitations, of individual tragedy and collective response. At the molecular level, Alzheimer's represents the fragility inherent in complex systems. The human brain, with its 100 billion neurons and trillions of connections, achieves remarkable feats: consciousness, language, abstract reasoning, artistic creation, scientific discovery. Yet this complexity creates vulnerability. The more intricate the machinery, the more ways it can fail. Proteins must fold precisely. Cellular cleanup systems must function flawlessly for decades. Blood vessels must maintain integrity. Any of countless molecular processes can go awry, and the accumulated damage eventually overwhelms the system. The disease also illuminates the relationship between biology and identity. We are not simply bodies with minds attached. Our minds arise from the physical activity of neurons. When those neurons die, when connections dissolve, when protein tangles choke cells, the mind disappears. Alzheimer's forces confrontation with the material basis of consciousness, with the reality that self and personality depend on biological processes that can be destroyed by disease. This confrontation challenges philosophical and spiritual frameworks that separate mind from body. Many cultures and religions understand humans as having souls or spirits distinct from physical forms. Alzheimer's raises difficult questions. Does the soul remain when the mind is gone? Is the person still present when they cannot recognize loved ones or express coherent thoughts? Different traditions offer different answers, but the disease inevitably forces engagement with these questions. From an anthropological perspective, Alzheimer's disease tests human social bonds. Humans are unique among animals in the extent and complexity of our caregiving. We care for the severely disabled, the profoundly cognitively impaired, the elderly beyond functional capacity. This caregiving reflects values about human worth, about obligations to family and community, about what we owe each other. Societies that provide robust support for dementia care express these values differently than societies that leave families to struggle alone. The disease also reveals economic priorities. Resources devoted to research, care infrastructure, and caregiver support reflect collective choices about where to invest social wealth. Wealthy nations can afford extensive dementia research programs and sophisticated care facilities, though even they often underfund these efforts. Poor nations must make brutal choices between addressing dementia and countless other urgent needs. These disparities create a global inequity, where place of birth dramatically affects the experience of developing Alzheimer's. Scientific progress on Alzheimer's has been simultaneously remarkable and frustrating. Researchers have achieved detailed understanding of disease mechanisms, mapped the progression of pathology, identified genetic risk factors, and developed methods to detect disease years before symptoms appear. Brain imaging can visualize plaques in living people. Blood tests can detect disease-related proteins. Yet, this scientific knowledge has not yet translated into effective disease-modifying treatments. Multiple therapeutic approaches have failed in clinical trials. Drugs designed to clear beta-amyloid plaques have mostly proven ineffective, raising questions about whether amyloid is truly the critical target or whether interventions come too late to reverse extensive damage. Approaches targeting tau tangles, inflammation, and metabolic dysfunction are under investigation. Some recently approved medications show modest benefits in slowing decline, offering hope while falling far short of a cure. The difficulty in developing effective treatments reflects the disease's complexity. Alzheimer's is not a simple problem with a simple solution. Multiple pathological processes interact over decades. By the time symptoms appear, extensive, irreversible damage exists. Trials require years to demonstrate effects on slowly progressing disease. Recruiting participants, measuring outcomes, and proving safety and efficacy present enormous challenges. Prevention represents perhaps the most promising frontier. Understanding risk factors, cardiovascular health, education, social engagement, physical activity, diet, sleep quality enables identification of modifiable behaviors that might reduce risk. No intervention guarantees prevention, but population-level approaches promoting brain health throughout life could reduce dementia incidence. The challenge lies in implementing these approaches across diverse populations and sustaining behavioral changes over decades. The story of Alzheimer's is also a story of advocacy and community response. Families affected by the disease have organized to raise awareness, fund research, improve care, and influence policy. These advocacy efforts have increased research funding, advanced legislation supporting caregivers, and reduced stigma. They exemplify how collective action can address diseases that individual families cannot tackle alone. Cultural representations of Alzheimer's in literature, film, and media have evolved from near invisibility to frequent depiction. These representations shape public understanding, sometimes perpetuating stigma through frightening portrayals, sometimes building empathy through humanizing narratives. The stories told about Alzheimer's influence how society responds, whether with fear and avoidance or compassion and support. Looking forward, multiple scenarios are possible. Continued research may yield disease-modifying treatments that prevent or dramatically slow progression. Early detection combined with preventive interventions might reduce incidence. Improved care models could substantially enhance quality of life even without curing disease. Alternatively, incremental progress might continue, with modest improvements rather than breakthroughs. The aging global population guarantees that Alzheimer's will remain a major challenge for decades. Ethical questions will intensify. As diagnostic capabilities improve, more people will learn they have early disease or elevated risk years before symptoms appear. How should such information be used? Should people be informed of predictive test results? How to prevent discrimination in employment and insurance? If effective preventive treatments emerge, who gets access? These questions have no simple answers. The Alzheimer's story ultimately returns to where it began with the aging brain, proteins misfolding, cells dying, and memories dissolving. The disease emerges from the deep biology we inherit through evolution, shaped by genes selected over millions of years when few humans lived long enough to develop dementia. It manifests in individual brains through molecular processes occurring over decades. It impacts families and societies through the demands of caregiving and resource allocation, and it challenges humanity to respond with science, compassion, and social support. In the end, Alzheimer's disease is a human disease in the fullest sense, arising from human biology, revealed by human longevity, addressed through human science, experienced within human relationships, and confronted by human societies making choices about care, research, and values. The pathology may be molecular, but the meaning is fundamentally human. As our species continues aging, as science advances, as societies evolve, the story of Alzheimer's will continue to unfold, shaped by our collective decisions about how we care for the most vulnerable, how we allocate resources, and what we believe we owe each other when disease takes so much but leaves the body behind.